DNA replication. DNA is responsible for one of the most essential functions of living organisms, the storage and transmission of hereditary information. A human cell normally contains 46 structural units called chromosomes. And you see the chromosomes here. We have our 46 structural units or 23 pairs. Now, each chromosome contains one molecule of DNA tightly coiled about a group of small basic proteins that we call histones, and individual sections of DNA molecules make up genes, which are the fundamental units of heredity. Now, if you remember from the first video in this chapter that each gene directs the synthesis of a specific protein, well, the number of genes contained in the structural unit of an organism varies with the type of organism. For example, a virus, the smallest structure known to carry genetic information, is thought to contain from a few to several hundred genes. A bacterial cell like E. coli contains about 1,000 genes, whereas a human cell contains about 25,000 genes. The process by which an exact copy of DNA is produced is called replication. It occurs when two strands of DNA separate and each serves as a template or a pattern for the construction of its own complement. The process generates new DNA double-stranded molecules that are exact replicas of the original DNA molecule. The two daughter DNA molecules have exactly the same base sequences as the original parent DNA. And so you can see here in this schematic that we have our original parent DNA, and then it undergoes replication, and we end up with a pair of daughter DNA molecules. Now, the two daughter DNA molecules have exactly the same base sequences as the original parent DNA. Moreover, each daughter contains one strand of the parent and one new strand that is complementary to the parent strand. This process is called semi-conservative replication. Now, the DNA replication occurs in three steps. In step one, which is the unwinding of the double helix, replication begins when the enzyme DNA helicase catalyzes the separation and unwinding of the nucleic acid strands at a specific point along the DNA helix. In this process, hydrogen bonds between complementary base pairs are broken, and the bases that were formerly in the center of the helix are exposed. The point where this unwinding takes place is called a replication fork. In step two, we have the synthesis of DNA segments. DNA replication takes place along both nucleic acid strands separated in step one. The process proceeds from the 3' end toward the 5' end of the exposed strand, which is what we refer to as the template. Because the two strands are antiparallel, the synthesis of new nucleic acid strands proceeds towards the replication fork on one exposed strand and away from the replication fork on the other strand. The new or daughter DNA strands form as nucleotides complementary to those on the exposed strands. They're linked together under the influence of the enzyme DNA polymerase. The daughter chain grows from the 5' end toward the 3' end as the process moves from the 3' end of the exposed or template strand toward the 5' end. As the synthesis proceeds, a second replication fork is created when a new section of DNA unwinds. The daughter strand that was growing towards the first replication fork continues growing smoothly toward the new fork. However, the other daughter strand was growing away from the first fork. A new segment of this strand begins growing from the new fork, but is not initially bonded to the segment that grew from the first fork. Thus, as the parent DNA progressively unwinds, the daughter strand is synthesized as a series of fragments that are joined together in the third step. And so in step three, closing the Nix, one daughter DNA strand is synthesized without any Nix, but the Okazaki fragments of the other strand must be joined together. An enzyme called DNA ligase catalyzes this final step of DNA replication. The result is two DNA double helical molecules that are identical, each of which consists of one original parent strand and one new or daughter strand. Observations with an electron microscope show that the replication of DNA molecules in eukaryotic cells occurs simultaneously at many points along the original DNA molecule. These replication zones blend together as the process of replication continues. This is necessary if long molecules are to be replicated rapidly. For example, it's estimated that the replication of the largest chromosome in the fruit fly would take more than 16 days if there were only one origin of replication. Research results indicate the actual replication is accomplished in less than 3 minutes because it takes place simultaneously at more than 6,000 replication forks per DNA molecule. The figure that you see here shows a schematic diagram in which replication is proceeding at several points along the DNA chain. Knowledge of the DNA replication process led scientist Kary Mullis in 1983 to a revolutionary laboratory technique called the polymerase chain reaction, or PCR. The PCR technique mimics the natural process of replication in which the DNA double helix unwinds. As the two strands separate, DNA polymerase makes a copy of each using the strand as a template. To perform PCR, a small quantity of the target DNA is added to a test tube along with a buffered solution containing DNA polymerase, the cofactor magnesium chloride, which is needed by the polymerase to function, the four nucleotides, or the four building blocks, and primers. The primers are short polynucleotide segments that will bind to the separated DNA strands and serve as starting points for new chain growth. The PCR mixture is taken through three-step replication cycles. Number one is denaturing. So this is where it's heated from 94 to 96 degrees Celsius. And we do this for 1 to several minutes to unravel or denature the DNA into single strands. This replaces the DNA helicase used to unwind the DNA in nature. Step two is annealing. So we cool the tube to 50 degrees Celsius to 65 degrees Celsius for 1 to several minutes, during which the primers, which are complementary to the target DNA sequence, hydrogen bond to the separate strands of DNA. In step three, extension, the tube is heated to 72 degrees Celsius for 1 to several minutes, during which time the DNA polymerase synthesizes new strands of DNA. And so each cycle doubles the amount of DNA. Following 30 such cycles, a theoretical amplification factor of 1 billion is attained. And thus, almost overnight, PCR became a standard research technique for detecting all manner of mutations associated with genetic diseases. PCR can also be used to detect the presence of unwanted DNA, as in the case of a bacterial or viral infection. Conventional tests that involve the culture of microorganisms or the use of antibodies can take weeks to perform, and PCR offers a fast and simple alternative. The ability of PCR to utilize degraded DNA samples, and sometimes the DNA from single cells, is of great interest to forensic scientists. PCR also has permitted DNA to be amplified for some unusual sources, such as extinct mammals, Egyptian mummies, and ancient insects trapped in amber.